So if viruses aren't alive, how do they reproduce? So to talk about this, I'm going to go into two separate examples. So first, we're going to talk about bacteriophage T4, which is a virus that does not infect people. It infects gram-negative bacteria, specifically E. coli. It does illustrate a number of important points in virology. It's got a genome made of DNA, double-stranded DNA. It's got a capsid that's icosahedral. It's made up of capsamere proteins that give it this regular geometric shape. It's non-enveloped, so there's no phospholipid bilayer membrane. Some other important structures are tail fiber proteins, spider leg-looking things that are used to attach to the host, and a tail sheath proteins that it uses to inject its genome into the host. You can see that structure in the electron micrograph of that virus attached to the host cell. We're also going to talk about influenza virus. Influenza does infect humans. It causes a disease that we call the flu, but it also infects other animals, including birds. It's got a genome made of RNA. In fact, there's 8 separate single-stranded RNA chromosomes that encode all of influenza's genes. It's got a capsid that has a complex structure. The protein that is the capsamere is called M1, and it's enveloped, so there's a phospholipid bilayer membrane on the outside of the capsid. Some other important structures in the virus include 2 spike proteins called hemagglutinin, or HA, that's used to attach to the host sialic acid receptor, and neuraminidase, or Na, that enables the virus to release from the host. It's got an important viral enzyme on the inside of the capsid called RNA-dependent RNA polymerase that's needed to replicate the RNA genome. So what does it mean to be an acellular obligate parasite? So, viruses don't possess all of the tools they need to reproduce. They have to hijack the host cell. Viruses don't have metabolism. The host cell has to provide all the energy to make viral chemicals. Also, viruses can't produce their own proteins. They lack ribosomes, and they need to have the host cells translate all of their genes. Because they lack all these things, virus genomes are much smaller than cellular life. Human genome is about 3 billion nucleotides long. E. coli's genome is about 5 million nucleotides. Influenza A has a genome that's 15,000 nucleotides in length. So, orders of magnitude smaller than the human cells that they infect. Because viruses have a much different molecular biology than their host cells, they sometimes need virus-specific enzymes in order to reproduce, including the RNA-dependent RNA polymerase that can replicate RNA genomes of RNA viruses. Another important concept to understand is that viruses have been evolving with their hosts for billions of years. There are probably viruses that infect every single type of cell on Earth. And in this billion-year complex relationship, viruses have evolved many tricks to hijack host cells, and those host cells have evolved many defenses to protect themselves from viral infection. Because of this complex host-parasite relationship, viruses are usually adapted to infect only certain host organisms or host cells. They have a host range, and this is called viral tropism. So, viruses that infect multiple hosts usually only infect evolutionarily related hosts. Bacteriophages don't infect human cells. They can maybe infect other bacteria. Some animal viruses can infect humans, but some can't. There are 5 general steps for virus reproduction. First, attachment. The virus has to attach a susceptible cell that it encounters by hooking on to a host cell receptor on the outside of the cell surface. Next, the virus has to get inside the host. It has to go through the cell membranes and the cell wall. We call this step penetration. So different viruses have different strategies to get inside. Next, all the parts of the virus have to be made inside of the host cell. So virus genomes have to be replicated, and virus proteins need to be produced and translated. We call this step biosynthesis. Then those parts of the virus have to be assembled into whole virus particles. Finally, those new virus particles have to be released from the host cell in order to attach to and infect another host. This step 2 can happen in different ways depending on the virus. Some viruses lyse the cell to destroy it, and some viruses exit through exocytosis. So the first example we're going to talk about is bacteriophage T4. Bacteriophages, or phages for short, are viruses that infect bacteria. T4 infects E. coli. A pretty simple structure: protein and DNA. The capsid is made of protein along with a tail, and the genome is made of DNA. They illustrate a number of important principles of viruses, so they're a good example to talk about, but we're also increasingly recognizing the importance of these viruses. One, because phages kill bacteria, and so they can potentially be used to treat bacterial infections. They also probably play a really important part in our microbiota by affecting the bacteria that live inside of us. So the phage life cycle can happen in 2 different ways. In the lytic cycle, which we will talk about shortly, the virus enters the host cell and makes new viruses, releases those viruses from the cell. Also, phages can reproduce through a lysogenic cycle, where the virus enters the host cell, makes its genetic information part of the host genome in a step we call integration, and then the host cell reproduces the viral genome when the host cell divides. This continues until the virus genes are re-expressed. I'm not going to talk about the details of the lysogenic cycle, but I would like you to read over it in the textbook, because this cycle is analogous to the way a human virus, HIV, reproduces and exists inside of our cells. So, a couple important terms: lytic phages are phages that only replicate through the lytic cycle, and temperate phages are phages that can replicate in both ways, lysogenic and lytic. One important concept too: phages transmit genetic material during their reproduction. Sometimes the bacteria actually benefit from this by acquiring useful genes. And so this is a type of horizontal gene transfer that we call transduction. So the bacteriophage lytic cycle happens like this. First, the bacteriophage attaches to the surface of the host cell using those phage tail fiber proteins. Then the virus has to get inside the cell. And importantly, the only part of the virus that goes inside is the phage DNA. The virus injects the DNA using its tail sheath proteins. The DNA contains all the information to make all of the virus proteins. So once the DNA is inside the cell in step 3, the host cell is tricked into transcribing those viral genes, translating the viral proteins, making new virus particles, and replicating the DNA genomes. In step 4, those viruses are assembled in a step that's called maturation here. Those fully assembled bacteriophage particles are then able to go out of the cell and infect new hosts. This step is what we call lysis for bacteriophages, because those phages lyse or destroy the host cell membrane and wall, killing the host cell in the process. Our animal virus example is a little bit different. We're going to talk about influenza. So influenza is an RNA virus. It's got 8 RNA chromosomes that make up its genome. It's also got 2 important spike proteins, hemagglutinin and neuraminidase, H and N. It's also an envelope virus, so there's a phospholipid bilayer on the outside of the capsid. Influenza virus causes a disease that we call the flu, which is characterized by a number of symptoms that you're probably familiar with: runny nose, sore throat, coughing and sneezing, fever, muscle aches, and occasionally vomiting or gastric symptoms. Influenza reproduces much like the bacteriophages, but also a little bit different. So first, the virus attaches to the host cell, and it does this through those hemagglutinin spikes attaching to cell host-specific receptor proteins called sialic acid. Once the cell is attached to the outside, once the virus is attached to the outside of the cell, then the virus induces the cell to phagocytize the virus, and bring it into the cell. So the virus tricks the host cell into undergoing this normal cell process, phagocytizing the virus, in order to get inside. Once it's inside, that endocytic vesicle that the virus has been brought into bursts, and the virus is uncoated, releasing all those contents into the cell. Then, those viral RNAs can go into the nucleus, where they are replicated, and also the viral genes are turned into mRNAs, where they are exported into the cytoplasm and translated into virus proteins. Now, it's important to note that the viral RNA needs to be replicated through a viral-specific enzyme called the RNA-dependent RNA polymerase. Only the virus has this gene and not the host cell. Once new viruses are assembled, then they are released from the cell through exocytosis. So similar to the way they get in, those viruses trick the host cell to exocytize or package those viruses in the phospholipid bilayer membrane and export them through the cell. So the influenza envelope is actually acquired through the host cell during this exocytosis process. So, viruses are obligate intracellular parasites, and so some of the tricks that we use to study these viruses in the lab are a little bit different than we do with bacteria. So when we grow bacteria on agar plates, viruses can't grow on their own. They need host cells to reproduce. So in the laboratory, we can use two different systems to grow and study viruses. We can use model organisms like mice to grow and study viruses, what we call in vivo, or in the living organism. Or we can use cultured cells to grow viruses in vitro, a Latin term that means in the glass. We also use chicken embryos or fertilized eggs that can grow many viruses in vivo and are used to, for example, produce influenza viruses for vaccines. We also use cell lines such as HeLa cells. So HeLa cells are one of the commonly used human cell lines for virology research and a number of other important human research subjects. So these cells grew from tissue taken from a cancerous tumor from a woman, Henrietta Lacks, in 1951. These cells were taken without Ms. Lacks's consent. And I'd like you to read the Ion Ethics in Chapter 6.3 to learn a little bit about this. If you're interested in after that, you can also read an excellent book about Henrietta Lacks's life and the research that's come from her cells. And also, the story of her life is going to be in an upcoming movie produced by Oprah Winfrey. So, on the right, you can see how we grow some of these viruses in the lab. What's shown is a plate with a plaque assay. There's a layer of human cells grown on the bottom of this dish, and they're stained purple with a dye to make them visible. The plaques are the little holes that you see in those purple layers. Those are where the viruses have killed the host cells during the infectious process. So these plaques are a little bit like the colonies that we see on bacteria plates. They started from one virus that infected a host cell and then killed all of the surrounding cells to make a little hole in the cell layer.